Hey everyone. So I'm going to go through the other part of protein synthesis today, and that is called translation. Um, Before I do that, I'm just going to go over transcription again pretty quickly, just so that we can see the whole process playing out. So I'm just going to go back in the notes. There we go. So remember, in transcription, let me just move me out of the way there a bit. In transcription, DNA gets unzipped here in the middle. Then there's this enzyme which grabs the RNA base pairs and it copies the DNA strand. So here on the bottom, this is the DNA feed, and then this red one here is the RNA strand, and you can see that it's grabbing the opposite base pairs. So with T, there's an A. Remember, for RNA, there is no T, so it makes a U. And then C goes with G, just like it usually does. And then the last step is it takes that mRNA and it leaves the nucleus. What it does is it's going to go to the ribosome, and that's what translation is. So once the mRNA leaves the nucleus, find a ribosome where the R to begin the process of translation. So the mRNA, it attaches itself to the ribosome, and then the ribosome is going to read the mRNA in groups of 3. So 3 letters, like in 3-letter chunks, basically. So these are called codons. We're going to be talking about those again in a little bit. So here's the diagram. You can see here at the bottom, there's the mRNA, and it's inside the ribosome here. You can see that these groups of 3 are called the codons. Then what's going to happen is that there's going to be this, the other RNA, the tRNA, and what it's going to do is it's going to grab the opposite letters of the mRNA. So just like how in DNA the letters were opposite, in the tRNA and the mRNA, those letters are also going to match up opposite. It's always going to be the same opposite letters. But what this tRNA does is it also brings with it, let me just move me there, what's called an amino acid. So these codons, they actually code for different amino acids. Acids are going to grow into a long chain depending on how long the mRNA strand is. And then this chain is going to combine to make proteins. So that's the first 2 steps. The mRNA attaches to the ribosome, and it is being read into codons. And then what's going to happen is the ribosome, to find that amino acid that matches the mRNA codon. And actually the tRNA group of 3 is called an anticodon. Again, we're going to be looking at that. So this is, you can see the tRNA with its amino acid attached on. So remember actually, this picture is a little bit off. G should be with C and A should be with U. This picture is backwards. Sorry about that. But yeah, so the tRNA brings back that amino acid. And then what happens is, like I said, the amino acids are going to connect together, creating a strand. And that strand is a protein. So that is what protein synthesis is. Okay, here's another picture. You can see here, there's the chain. It's going to combine into a protein. So I'm going to be doing another example of basically how this stuff matches up. We're also going to be talking about codons either tomorrow or later next week. I'm not too sure yet. But this is going to primarily be what we're going to be working with for the rest of the unit. I'm also going to post a couple of extra videos that can explain this stuff a lot better than I ever could. But if you have any questions, feel free to email me or Monique. And until next time, have a good one, guys. See ya.